So some amino acids are essential, okay? Of the 20 amino acids that are used to build the proteins in the body, 11 can be made in the body, but the other 9 are actually essential that we eat from our diet. And so these are histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. Say that 10 times along with Rumpelstiltskin, and you will be good. Just kidding. Okay, so basically we have to have a complete diet, and so complete proteins such as eggs, milk, meat, fish all contain the essential amino acids, and incomplete proteins from plants such as grains, beans, and nuts are deficient in one or more of the essential amino acids. So that's why a well-balanced diet is very important. So amino acids as acids and bases. When amino acids with a positive or negative charge, it's an overall neutral charge. This is said to be an isoelectric point, or pi. So this would be an example of the ball and stick model of glycine, and its pi is 6. So what we're going to learn now is to draw the condensed structural formula for an amino acid at pH values above or below its isoelectric point. So what is the isoelectric point again? Well, it is the pH at which the charged groups on the amino acid are balanced, and therefore the amino acid is neutral. Because if we have a positive charge and then we have a negative charge, they balance out to be neutral. Okay, an amino acid can exist as a positive ion if the solution is more acidic, which would mean the lower pH, than its pi. And it is a negative ion if the solution is more basic or higher pH than its pi. Okay. So looking at ionized forms of amino acids, the pi values for nonpolar and polar neutral amino acids are from pH 5.1 to 6.3. And alanine here that's pictured, okay, this has a 0 overall charge at its pi of 6 with its carboxylate anion, the COO minus, and the ammonium cation. Okay, so that's the plus and the minus, so they equalize out. Alanine is going to add a hydrogen to the carboxyl group. Okay, so you can see here, alanine at the pH of 6 is COO minus versus COOH. Okay, so it adds this hydrogen to that carboxyl group when the solution is more acidic than its pi. So that means, so if the pi is 6, then when it's more acidic, that would mean below pH of 6. So it's really silly how I remember this, and you guys are all super duper smart, so you probably already know this, but I remember the acidic numbers are lower than 7 because people back in the day used to drop acid. Okay, so if you're dropping below 7, then that means you're going to be acidic. Okay, so if we're normally at 6 and then we drop below it, then that's going to be what happens here. Okay. All right, so at a pH higher than 6, then the NH3 group loses a hydrogen and it forms the amino group that has no charge. Okay, so this is the NH3 plus, and then it drops one of those hydrogens to make the amino group. So because the COO minus group has a -1 charge, then alanine has an overall negative charge of -1 at a pH that's higher than 6. So, looking at a fun little diagram, what do we know? Well, we can add a hydrogen, and that would give us a positive ion and a lower pH. This would be when we're at our pi value, and then if we get to a higher pH, or more basic, okay, then we are going to have the negative ion. So you see that we dropped a hydrogen here from versus there to there, okay? So here we're adding a hydrogen, and it's giving it a positive charge, right? This one we're dropping a hydrogen and actually the hydrogen from the carboxyl group. All right, so looking at this, which structure represents alanine at a pH above its pi and alanine at a pH below its pi? Pause me and come back. All right, the second one is going to be representing alanine at a pH above its pi, where number 1 here is alanine below its pi. All right, let's try another one. So consider the amino acid leucine with a pi of 6. So at pH of 3, how does leucine change? And if we change the pH to 9 to more basic, how does leucine change? Pause me and come back. All right, well, at a pH of 3, because the pH of 3 is more acidic than the Pi value of 6, then the COO minus group gains a hydrogen to give it the COOH. Then the remaining NH3 plus gives leucine an overall positive charge of 1. Where at pH 9, how does it change? Well, because pH 9 is more basic and it's above the Pi of leucine, then the NH3 plus loses the hydrogen to give it NH2. Then the remaining COO minus gives leucine an overall negative charge of -1. All right, great job, you guys.